Hello, everybody, and welcome to another episode of General Biology 1. Today, we're going to talk about chloroplasts, what they look like, and how they work. But before we get into that, when growing up, I was a bit of a Star Wars fan. I can't say I was huge into it like some other people, but I always wanted a lightsaber. And if you remember the lightsabers, they're basically this point of this sword of light that kind of ends at a point and apparently has some mass to it where you can kind of hit each other, right? And it makes that vroom, vroom, vroom sound. So, got me thinking, could you ever produce a real lightsaber? What do you think? Will it ever be possible to have your cats settle their differences in this very entertaining way? Well, believe it or not, there are people working on it. So, there was a YouTube sensation video a while back with this guy who had made his own lightsaber. And he used plasma. And here it is in action, cutting through someone. Not quite as quickly as effectively, but it's basically like a super hot blowtorch. And it was made with plasma. And you've probably heard of plasma before. Some people call it like the fourth state of matter. And in its simplest form, plasma is basically a gas that has become ionized. And if you remember what ions mean, it means that something's losing and or gaining an electron. And basically, if you take gas under high amounts of energy, it could actually cause nuclei or atoms and to shed their electrons, essentially leaving them as these protons and neutrons with a high energy state. So they're just releasing all these electrons, right? And it creates this incredible heat and an electromagnetic force. And when this happens, we tend to think of it as sort of under astronomical conditions, something that you might see inside of the interior of a nebula or something like that. Um, but essentially, adding energy will cause an atom to lose its electron. Now, at this point, you're probably thinking about, I thought we're talking about chloroplasts. What's going on? Believe it or not, there is a connection. Just remember, with enough energy, an atom can lose its electrons. So, we were talking about photosynthesis, and photosynthesis is basically split up into two parts. You have the photo part, which is essentially something that occurs in light, and when light is available, a series of reactions work to break down dihydrogen oxide, otherwise known as water, and to break it down into essentially an oxygen molecule, and oxygen O2 being the end product of the light reaction. And the name light reaction tells you that light is involved. And if you recall, light hitting a water molecule in the right time, in the right way, can cause a dissociation of those oxygen molecules, right? What we called photolysis. So water and light go into the light reaction, oxygen is produced as a byproduct. But if you recall, there's other parts to photosynthesis, including the making food part. And that occurs in the Calvin cycle, which we think of as the synthesis part of photosynthesis. And in the Calvin cycle, what you're basically doing is you're taking carbon dioxide, which is floating around, you're fixing it, you're taking it from being this gas floating around that is essentially not usable by any living thing, and then turning it into a form that through a set of metabolic stages will eventually produce a sugar, right? And this is the so-called photosynthesis, the plants making their own food, what makes them an autotroph. This is something that can occur in the dark, so you don't need light for it. What you do need, however, are other sources of energy, in this case, the chemical energy that comes out of the light reactions. So ATP, you'll need that. And you'll also need sort of a high energy molecule, these electron shuttle acceptors, or electron acceptors or shuttles, like NADPH. And NADPH is used in the Calvin cycle, and then both NADPH, essentially loses that hydrogen, right, or becomes oxidized, leaving only NADP plus. And ATP gets used, leaving behind ADP molecules as well as inorganic phosphates. And those cycle back into the light reaction. So there's that connection there, right? So if you look at the photosynthetic equation of water plus CO2 plus energy in the form of light producing oxygen and sugar, It's basically occurring because of these two different sets of reactions. So today we're going to talk a little bit about sort of how this whole thing plays out. So again, the light reaction, you're taking light energy, right? The energy in light, which we'll talk about today, and you're turning into a chemical energy, right? What's going to happen is that light is going to photolyze water. And in the process of doing so, water is going to get oxidized, meaning that it's going to lose its electron. As it loses its electron, right, that electron is then going to be shuttled through something called the electron transport chain, or the ETC. And this is all occurring within the thylakoid. Right? So again, the thylakoids are those pancake stacks that are present inside of a chloroplast. And in the process of doing so, there's going to be energy transfer there. And part of that energy transfer is going to be used to make ATP, a process called photophosphorylation, which we'll get to, and then also to make those electron shuttles, that NADPH. So that's the light reaction in a nutshell. On the flip side, in the Calvin cycle, this is occurring in the dark. You don't need light. And you're, again, fixing carbon, right? Sometimes it's called a dark reaction as a result of this. You're fixing carbon, and you're going to use the products of the light reaction to do so. The end goal here is to take the carbon molecule, or the carbon atom, and carbon dioxide, and use it to produce a carbon sugar, all right, or a carbohydrate. So what part of the chloroplast is the light reaction occurring in, and what part of the chloroplast does the Calvin cycle occur in? Well, if you recall your basic chloroplast structure, your chloroplast is a organelle located inside of autotrophic plants and other things as well. And the chloroplast is basically kind of just a sac, really. It's got two membranes, an outer and an inner, and it has a set of these thylakoids that are organized into stacks, like a pancake stacks. And the thylakoid is each individual pancake. A stack of pancakes makes a granum. And then all of these pancake stacks are immersed in a fluid, otherwise known as a stroma. So the light reactions are occurring in the green part because you need that green chlorophyll. We'll talk about that. And those are located in the thylakoid, and specifically in the thylakoid membranes. Whereas the Calvin cycle is actually a process that occurs outside of the thylakoid in the stroma. So if you take a close look at a leaf and you do a cross-section and you look at it at the cellular level, you'll notice two things. One, that there's a specific organization to how a leaf looks, right? The outer versus the inner parts of a leaf. If you look at it in a cross-section like here, you have the outer and lower epidermis, right? These are usually specialized cells, and you'll notice that those cells don't tend to have a lot of chlorophyll. They don't tend to have a lot of chloroplasts. The chloroplasts are primarily concentrated in what they call the mesophyll cells, right? And you have the palisade mesophyll cells near the upper epidermis that are kind of sort of organized in sort of top-down kind of fashion. Then you have the other layer near the lower epidermis, which is the spongy mesophyll. So palisade mesophyll, spongy mesophyll. These are the things that have the chloroplasts. And again, those chloroplasts have thylakoids in them that are organized in stacks called a granum. So inside the interior of a thylakoid, you call that the thylakoid space, and each thylakoid is separated from the other thylakoids in a granum by the thylakoid membrane. And that membrane is important, and we'll get to that. That's the site of where everything cool is happening. But here's a question. What is it that makes chloroplasts green exactly? Well, I'm sure some of you said chlorophyll, hoping. And you would be correct, but partly correct. See, chlorophyll is green, but the reason it's green is because of the way it interacts with light, as well as the way that we see it. So this is a good segue into talking about light and what it means, right? Everybody knows what light is, but when asked and pressed to give a strict definition, it's actually kind of hard. Light is basically particles. You don't see them, but they're these particles called photons that are moving through space, right? They're moving through the air. And they move in clusters, if you will, right? So you'll have areas, if you take a certain amount of space, for example, right, a certain volume, You could have a lot of particles in there, or you can have few particles in there. And if you have a lot of particles in there, and you know these are kind of set up where you'll have a box of particles, a little bit of space with less particles than another box of particles. That distance between those two boxes of particles, right? That would represent essentially a wavelength. Now, what do I mean by that? Wavelength. Well, wavelength is basically the distance between clusters of high density photons, right? So as a result, we tend to think of wavelength, abbreviated by the Greek letter lambda, as basically being an indicator of how much energy is available. So let's say you have a 3 foot or 1 meter distance, right? And let's say you have in that 1 meter distance, you have two clusters of high density photons, right? And then you have another meter of distance somewhere else. And in that meter, you have 5 clusters of photons. Well, all else being equal, the one with the shorter wavelengths between the clusters, i.e., the more clusters you have in a meter, the more energy there is because there's more photons in there. So the shorter the wavelength, the higher the amount of energy. So we know about wavelengths a little bit because we talk about them and we refer to them all the time. So when we talk about light, often we're talking about something that looks like this, the rainbow, which is basically our interpretation of the visible spectrum, right? This is what we call visible light. It ranges, Roy G. Biv, right? You go from red all the way down to a violet color, right? But that's only the part that we're able to see with our human eyes. If we were some other kind of animal, we can see more, right? Some animals, like birds, can see into the UV. Some, like pigeons, can even see a little bit into the IR, right, or the infrared, as opposed to the ultraviolet. So what we see, us humans being as arrogant as we are, we call it the visible light spectrum. Now, within that visible light spectrum, what determines the color is basically the wavelength, right? And then the shorter the wavelength, the higher the amounts of energy. So blue lights, purple lights, UV light, which can burn you very quickly if you're at the beach and you're not wearing sunscreen, right? X-rays, these things are high energy things because they have very short wavelengths. They pack a lot of photons into the same amount of space. As opposed to, say, lower wavelength things or longer wavelength light, which has lower amounts of energy. They can transmit farther. That's a different thing. Won't get into that. But there's lower amounts of energy. So infrared is very low amounts of energy. It's microwaves. Microwaves, you think, oh, microwave heats stuff up really quickly. It's because it actually interacts with water. So if something has water in it, the microwave will heat it up quickly because that water is able to absorb that particular wavelength, right? Microwaves. Fun fact, take an ice cube straight out of the freezer, put it on a plate, put it in the microwave, and you'll see it takes a while for it to actually melt, right? Because there isn't any liquid water there for it to work on yet. As soon as there's some liquid water, it'll start cooking. So, fun fact, try it out. You'd be surprised. The bottom line is, we think of light as wavelengths, but we're really talking about photons. Each of those photons having a specific amount of energy. So why are plants green? Why are leaves green? Well, 3 things can happen to light when it hits something. It could either be reflected, it could be transmitted, or it can be absorbed. Light that is reflected bounces off of the object and comes back, right? Transmission simply means that it gets through the object. And sometimes as it goes through the object, it might change, it might shift its wavelength a little bit, right? And this is an example of refraction. Or it can actually be absorbed. And what we mean by absorbed is the energy is actually hitting something that's going to take that energy and hold on to it, right? It's basically going to use that energy. So why are leaves green? Which of these 3 are at play there? Well, you should have guessed that leaves are green because they reflect back green, right? So So they absorb and transmit all the other colors, but green gets reflected back. And because that gets reflected back, that's what we see. Now, which of these processes are what photosynthesis is going to rely on? Which of these fates? Well, hopefully you guessed absorption, because we're going to use that energy for something else. So here is a picture, a graph showing wavelengths of light for three types of pigments: chlorophyll A, chlorophyll B, and carotenoids. And this is showing the absorption of light by these pigments located within a chloroplast. So this is what we call the absorption spectrum. So how much absorption is happening on the y-axis across the various wavelengths of light. So when you look at this graph, what would be the most effective wavelength for photosynthesis? Well, as I mentioned, we're interested in absorption, right? So it would be the peaks, basically, right? So you see the peaks, well, you get the majority of your peaks kind of in the blue-purple range of wavelengths. And you also have a secondary range kind of in the orange-red. Notice green, however. Green's right in the middle. You don't really have a lot there, do you? Right? Not a lot of absorption is happening there. That is actually where you get the reflection from. So you have chlorophyll A, chlorophyll B. Both of these chlorophylls are green. You probably already knew that. We'll get to carotenoids in a little bit. Because they're green, they're not absorbing at that wavelength. They're reflecting it back into your eyes. So that's what you see. Now, something like this can be confirmed using a more experimental approach. So here, they basically took a plant, they're measuring photosynthesis, and they're measuring the rate of photosynthesis, in this case, how much oxygen is being released. So if you remember, basically the more photosynthesis that's happening, the more water is turning into oxygen, basically. So more photosynthesis, more oxygen. So you look at across that range of wavelengths, well, you see the highest rates of photosynthesis in the same sort of peaks where you have the highest absorption. So that confirms it's the absorption at those wavelengths that are driving photosynthesis. Right? So this is an example where you have biochemical information from the pigments, but you're validating it with experiments using an actual plant. So here's another one, an unknown pigment right here. Across this range of wavelengths, an absorption spectrum, what would be the color of this pigment? So if you look at it, the lowest point on the lowest trough on this graph would be in the blue wavelength. So that means the least amount is being observed, or the least amount is being absorbed at the blue wavelength, i.e., it's probably being reflected back. So you're probably looking at the action spectra for a blue pigment. Now that said, the blue pigments are pretty rare. So how was photosynthesis figured out? So here's a cool thing. This is an experiment that was done quite a long time ago by someone named Engelmann. He's not important, but what is important is his experiment. So what he did was he took algae, and algae, of course, are photosynthetic, and they have chloroplasts and all that. And he basically shot a light through a prism. And what a prism does is it basically causes white light to turn into a rainbow. And it does so because of its shape. The light that goes through gets refracted. And depending on where it gets refracted, it might have to travel a different distance and a different angle. So every time it changes its angle, it shifts its spectrum over. So you end up with the right prism in the right location, with the right light hitting it, you'll produce the full visible spectrum as a result. So he shone this light through this prism and onto this algae. And then what he did was he had the algae basically in a solution or a setup with bacteria that were aerotactic or aerobic bacteria. So looking at this, right, you look at the setup here. What is the reason that the bacteria congregated where they did? What explains the pattern of the bacteria across the wavelengths of light by these that is hitting these chloroplasts? Okay, well, I'm guessing that what gave it away was the aerobic bacteria, right? Aerobic bacteria simply means that they depend on oxygen, right? There are bacteria that are anaerobic that don't need oxygen, but they need oxygen. And because they need oxygen, they're going to congregate in places that are high concentrations of oxygen. Now, since you're basically causing photosynthesis to happen by shining a light on these chloroplasts, well, you know that photosynthesis is a product of that light reaction is going to produce oxygen. So the places with the highest amount of oxygen will be the places with the highest rate of photosynthesis. So that's why the bacteria show up there, right? So that explains the distribution of the bacteria. Again, the green color, they're reflecting that back, right? So there's not a lot of photosynthesis happening there, so you don't see a lot of bacteria on the other side of that membrane. It's a pretty cool experiment, to be honest with you. Quite ingenious. Hats off to Engelman. Okay. So now that we know a little bit about light and we know about how light behaves, let's take a look at chlorophyll. So chlorophyll is an interesting molecule. It's made up of a porphyrin ring. So a lot of molecules have metals associated with them, and in this case, magnesium is the metal that kind of holds this ring together, right? So long story short, you know that those little corners, based on your organic chemistry knowledge, whatever that state of that might be, each of those corners represents a carbon, right? So you're looking at the carbon skeleton. And in the middle of the carbon skeleton is this magnesium. Now, the magnesium is important because it's the magnesium that is a metal, and it's the part that absorbs light. So you have this porphyrin ring, but you also have this other part, which is kind of hanging off it, which is a hydrocarbon tail. And if you think about a hydrocarbon tail, it's a lipid, right? So it's kind of similar to the phospholipids that go into the cell membrane. And there's a reason for this. Well, it's got a hydrophobic part that it can put into a cell membrane. In this case, the thylakoid membrane, right? So it's able to be inserted into the thylakoid membrane as a result of that hydrophobic tail. And the porphyrin ring is just kind of out there. Now, there are two types of chlorophyll. There's a chlorophyll A and a chlorophyll B. Up there, you can see the small differences in that one little corner represent that, right? So basically you have, either it's a methyl group or a carboxyl group, whatever. That small difference is enough to change it, right, and how it behaves. Most chlorophylls that we're going to talk about are going to be the chlorophyll A type because they're the ones that are found in plants. And specifically, there are two versions of the chlorophyll A that we're going to talk about, a P680 and a P700. These are the two versions of chlorophyll A. Why do you think they have these numbers associated with them? Well, hopefully you picked up on the fact that the only numbers we've used so far are wavelengths. So it's actually the wavelengths, right? These are kind of the peak wavelengths that they interact with, right? So P680 interacts with 680 nanometer wavelengths. P700 interacts with 700 nanometer wavelengths. Quick mention about the chlorophyll B. It's found in algae. Often it's not playing the primary role. It's what they call an accessory pigment, meaning that it's there to help gather light, help get energy, but not necessarily the main chlorophyll that's working. So they have these other accessory pigments as well, these carotenoids. And in nature, whenever you see something that's orange or yellow or reddish in color, it's usually because of these carotenoids. So whether I'm talking about plants, whether I'm talking about insects, whether I'm talking about birds, those sort of colors are often the product of these carotenoids. So here's two sort of more famous carotenoids, beta carotenes, which the name kind of should bring to mind. Oh, carotene, carrots, right? It gives us that orangish color, right? That sort of orangish reddish color. And then xanthophylls. Xantho means kind of yellow. So those kind of give us the sort of the light browns, the yellow, the blondish colors, right? So beta carotene and xanthophylls are two examples of carotenoids, which are pigments. So these accessory pigments, they do a lot of absorbing in sort of the blue-violet end of the spectrum. So they're basically kind of interacting with the same kind of light, that chlorophyll is. And there's a reason for this. What's the purpose of these pigments, you think? Well, the name says it all. You're basically getting a little more energy. So think of it as like a radar dish, right? A radar dish has the middle thing that actually sends the signal or receives a signal, but it has this big funnel that helps to funnel the signal in. So that's kind of how these are working. They're helping to get a little bit more energy and contribute to the system, right? They're not the main drivers, but they're there to help. They're playing an accessory role. So when I first got to Rollins, I was actually interested in cardinals. I was working on cardinals and doing a lot of research on cardinals. Um, I got tired of waking up super early to go catch them though. But I do like cardinals, and you probably know cardinals as being really, really red. Usually you're thinking about the male cardinal, right? Like the one from Angry Birds. You remember Angry Birds? My kids loved Angry Birds. So Angry Birds, cardinal, you look at the red coloration, that's usually the male we're talking about. The female, however, has a little bit of red, but is mostly this brownish color. So why do you think male cardinals are red and females are not? Well, the answer was kind of given to you before. Males have a lot of beta carotene that they put into their feathers, right? They actually are able to take that beta carotene molecule pigment and incorporate it into their feathers when they molt. Whereas females tend to have more xanthophylls in their feathers, right? So that's the difference between them. Now, carotenoids are in plants, so the way they get them is through diet. So a cardinal is feeding on the ground, feeding on seeds. Those are very rich in carotenoids. They then consume them. Now, here's the thing. A male cardinal has the enzymes necessary to take those pigments and incorporate them into their feathers and often convert them in the process, right? They convert them from being xanthophylls to beta carotenes that they could put into their feathers. Well, female cardinals are eating the same thing. So they just don't have the enzymes that that convert those carotenoids and put them into their feathers, right? And of course, there's a reason for this, right? Brightly colored males get more females. Females need to stay a little bit cryptic so they can hide when they're on the nest, that sort of thing. But what's really cool is that female cardinals, and this is actually something that a friend of mine used to study and was one of the first to kind of describe this, but females are actually doing the same thing with the carotenoids, but instead of putting it into their feathers, they're actually putting them into their eye. They're actually incorporating those pigments into their photoreceptors. And by doing so, they're better able to discern red colors. So males are trying to look as colorful and red as possible for a female. Now to us, every cardinal looks the same amount of red. But to a female, well, she's putting these carotenoids into her eye, so when she looks at males, she can actually tell very subtle differences in their plumage. Even though they're all red, they're all slightly different red. So she can pick her best mate as a result, right? Which is really, really cool. So same machinery, just used very differently between the two sexes. Really cool stuff. Happy to talk about it more, but another time. Okay, so now you have what we have here, which is your chlorophyll A, your chlorophyll B, and carotenoids all working together with a primary focus on that blue-purple sort of end of the spectrum, right, as being the predominant wavelengths we're interested in. Okay, so let's talk about photoexcitation. So what happens when a photon of light hits a pigment molecule? So a couple things. First, as you know, any sort of atom will have electrons orbiting around it, and in a typical situation, that electron is going to be orbiting at its particular energy state, meaning it's going to be on a certain orbit, right? Whether it's the valence shell, whether it's one of the interior orbits, right? And those electrons are there. That's where they're stable, and they have a certain energy state associated with that particular place. Now, sometimes when a photon of energy hits just right with the right amount of energy, it can actually cause an electron to jump from one orbital shell, to the next orbital shell. And we talk about this as going from a ground state to an excited state. And every electron and every molecule has a certain amount of energy necessary to do this with, right? So if a photon comes along and hits it, and it has that right amount of energy, it could cause it to get excited, right? So this is photoexcitation. A photon exciting the electron, making it jump from a ground state to an excited state. But it's a very unstable place to be at another orbital shell that you don't need to be at. So what usually happens during that state is you're going to have that electron basically come right back down to the ground state. And in the process of doing so, it emits two things. One, a photon, and usually that photon is a certain wavelength, and then also heat. So why heat? Let's start with that. Well, the reason for heat, you got to go way back to the second law of thermodynamics, right? This idea of entropy in a system. Entropy in a system, in a biological system, we're almost always talking about heat, right? So anytime there's a transfer of energy from one thing to another, there's some loss of heat associated with that. So you get that loss of heat. Okay. You also have this photon that's emitted. This photon is what we call fluorescence. So when something is fluorescent, the idea for true fluorescence, what that means is that that thing is releasing photons of its own, right? And it's releasing photons of its own and usually of its own wavelength. So question here says, where does this exact amount of energy come from? Well, it comes from the photon that hit it. So usually what will happen is if the right photon hits a particular molecule and it causes those electrons to jump and they come back down, the photon that it's going to emit is usually the same wavelength as the photon that it received. Right. So that's kind of a the thing about pigments, right? They often use them to study these prospects of these ideas of fluorescence. Now, if you take chloroplasts, they actually also fluoresce. And what they'll do is they'll produce a red color. Well, if they're in the dark and you expose them to UV light, they'll fluoresce. If you take chloroplasts that have been purified, i.e. they've been broken down and they've been removed from their membranes and everything, they'll do this autofluorescence when you expose them to UV light. So you give them UV light, right? So that's a certain wavelength. So that's, past the it's ultraviolet, it's past the violet end of the spectrum. But then what's going to happen is they'll fluoresce, and then when you let them go, right, they will start to release photons of their own. But those photons are going to be red. Well, while they're releasing red, they also are releasing heat. So between those two, that's basically all the energy that they initially absorbed, right, when they fluoresced in the first place. Okay. So, I like to end with these ideas of why this stuff matters, right? Moving forward, we'll talk more about photosynthesis and how chlorophyll is working, but chlorophyll itself really matters, and light absorption really matters. One of the concerns about climate change is a lot of algae are not going to be able to basically make it. So even though we need them to fix carbon and get it out of the atmosphere and into the oceans as some kind of product that could fuel a food chain, and they'll produce oxygen as a result. There's concerns that, especially if conditions become very anoxic, i.e. in places where you're going to have an awful lot of pollution that could potentially cause algae to not even be able to survive. Usually we associate algae with pollution, but they're actually not. They need really good places with high nutrients, right? They can damage an environment when they grow too much into an algal bloom, but they are part of a food chain, as opposed to, say, having anaerobic bacteria. And anaerobic bacteria in the ocean, many of them can also engage in, not anaerobic bacteria, aerobic bacteria. Many of them can also engage in soaking up light, and when they do, they use a different molecule. Instead of chlorophyll, they use something called rhodopsin. Now, rhodopsin is an important molecule in bacteria. It's also an important molecule in mammals. It's actually part of our photoreceptors. You can take one of my classes for that. But long story short, the rhodopsin is in bacteria is basically used by these bacteria to soak up oxygen, right? So they're taking in oxygen, but they produce CO2 as a byproduct. So they're acting like a heterotroph. They're not producing food. They're not producing energy. They're not producing. They're not taking away carbon dioxide. They're adding it to the system. So that's a big concern. Is just these two different molecules and how they interact could potentially cause big problems in the future, right? With warming temperatures and increased pollution. So chlorophyll matters just like everything else matters in the system. All right, so as we move on, we'll start talking about photosynthesis and how exactly it works. But until then, take it easy.